Okay, so I'm going to try and demonstrate how to build the DNA molecule using the paper. So I've already put this together. I just attached the two parent strands, so they're put together. They're basically the same strand twice, so it shouldn't matter which one you put on which side. So I'll follow the steps on your handout to go through this. So I've already done steps 1 and 2. I attached the two parental strands. So the top strand, it's kind of hard to see, but hopefully if you have this printed out, it shouldn't be too bad. So it goes 5' to 3', 5' to 3' over here. So remember, both sides, they run anti-parallel to each other. So, very first part. Turn that DNA print, the parental strand is wound up in a highly twisted double helix. So remember to be all twisted up. Really hard to do with paper, but that's okay. We can just pretend. So it unwinds due to DNA helicase. It's going to unwind it. And then DNA helicase is going to start to split this molecule right down the middle, breaking these hydrogen bonds in between each of the nucleotides. Really, really awkward angle here, so I can't say I've ever cut like this before. That's okay. So really, on the instructions, it says to start cutting towards the middle. So we start to see a replication fork going on over here. As you can see, these two strands are going to start to split. Here's that replication fork right here in the middle. This is going to continue down. Really, this process would happen to a point where, as this replication fork is splitting, DNA would start to get built over here on this end. And just follow down, and this would continue to get split, but since I only have two hands, it's really hard to do that. So I'm going to go ahead and just split the molecule all the way, but I'll try to keep it closer over on this end so we can still see the fork. So we'll just split the molecule all the way. Remember, this is DNA helicase breaking those hydrogen bonds. So I've split the molecules, I've split both sides. It should look something like this. So there's that replication fork, but here down on these ends, these two, maybe you can see them better if I hold it like this. These two ends, these sides are the ones that we're going to start to work on. So we'll lay them down. We have a top and bottom parental strand. To relieve stress of untwisting, we put little nicks in the backbones to help relieve that stress of being basically straightened out. So we just put little cuts in there. I'm not going to tape them back together, but they would get put back. So we'll attach some single-stranded binding proteins. So I have them out here, super teeny tiny. They'll just be bonding out to the outside. And again, really all these are going to do is just they're just going to make sure that these molecules don't come back shut. So here is one. There's one protein. I'll go ahead and attach the rest really quick. These I would just put on the outside just because if you put them on the inside of the molecule, like in here with this, where the fork is, you're going to end up blocking some things that you'll need later on. So I'll just put them on the outside part just because it makes things easier. Okay, thought I lost one. Okay, so there are all the proteins. They've been added to the outsides of both the strands. There they are. You can kind of see them there. Up here too. So we use topoisomerase to relieve some stress, added single-stranded DNA binding proteins to make sure that they stay open. So now, DNA primase, I didn't cut them all out, it's going to be building an enzyme, or a primer, sorry, it's going to be building the GU or GUGU primer. This primer is going to be added on the right side of the top parental strand. So this strand over here is going to be added to the right side So as you can see, there is a CA, which very conveniently pairs up with GU. They're going to be matched. Some people, I was noticing, added the GU over here like this. That's not how we built DNA. First off, the bonds are all wrong. Second, we never added anything to the DNA molecule. We're literally just copying it. So for this part, we're just going to pair it up right there on the bottom, just like that. So as you can see, they pair up very nicely and match up. So I'll add some tape there. So GU pairs up with CA. Now, we have our primer beginning. Kind of hard to tell, but this end up here is 3', so that means that this end down here has to be 5', and this one is 3'. So to move 5' to 3' on the new side, it's going to move this way. So we'll be building everything that way. If you remember, following that, our replication fork is opening up this way, and we are building our strand this way, so this is the leading strand. So as this fork opens up, as it's going, it's constantly opening, we're constantly able to add DNA the whole time. So this one, we're able to add long chunks of the DNA. So we have the 2 chunks of the leading strand. On this, you just need to find the parts that match up. So there is this end over here. It has the T and the A. So the A, T match up, GC, CG, those triple A's with the triple T's. So all that part is going to match up. Really, each nucleotide would be added one at a time. And again, it would be in the 5' to 3' direction. On here, it'd be really obnoxious to have to cut out a bunch of things. Even though I made you cut out a bunch of things anyway, but I didn't want to make it even more obnoxious than what it was. So there's that part. Here's more of the leading strand. It's just a continuation part of it. Again, these would just be being added, basically just one nucleotide at a time by DNA polymerase 3. It would just keep going. So there is our leading strand. This one's pretty much built. So we'll stop working with that one and put it to the side. Remember, it would still be going like this. This one would simultaneously be being built at the same time, but that's just kind of hard to do. So now, we just created the leading strand, so we're back on, step 15. So we still have the replication fork. This one is going to be our lagging strand, this new strand being built here. So we first need to start placing some primer. So again, that is DNA primase that makes this primer. It's going to lay down this GUAC near the right side. So we have near the right side of the strand that CATG is going to pair up with GUAC, match up with guac. So we're going to place that there. I'll add some tape. Now, lagging strand, reason it's the lagging strand is because it's built backwards. So again, this is our replication fork. Leading strand built this way, follows the fork as it opens up. This one, however, is going to be being built backwards. So we lay down a primer, 5 to 3' on this side goes that way. So it's moving away. So it's going to add just a little chunk over here. So it's TGT. So the matching chunk of DNA would have to be ACA. So there's that chunk. This gets laid down still by DNA polymerase 3. It's just having to work 5' to 3', which happens to be away from the fork on this end. So as this continues to open up, we get more room for another primer. I'll be real honest, I don't know which primer is next. Oh, I think I found it. UCCA matches up with AGGT. Hope you can see that. So this primer goes here. So primase makes another primer, lays it down. Now we have this chunk of DNA to fill. So again, it's working 5' to 3', so it's going to be working backwards towards the primers it's already made. So here we have this chunk. So boom, 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 boom. All of these DNA nucleotides get laid down. Tape them down as well. And then we continue to open up. And we have one more primer, AAAC. This one's easy because it matches up with the TTTG. Whoop, there we go. So we're just going to lay down this one. Again, DNA primase is the one building these and laying these down. That's its sole purpose. We have the one last chunk of DNA, fits perfectly right in here, which is going to be laid down. Again, as this was opening up, primer got laid down, had to build backwards. Constantly has to play catch up. So that's why this is the lagging strand. Oops. Okay, so that last chunk has been laid down kind of sloppily, but that's okay. So remember, all of these DNA bases have been complementary paired. They're all matched up. These chunks of DNA that were put in between all of these primers over here, these are the Okazaki fragments. So these chunks are, they're just stuck in between all of these primers. So we're done laying all of the DNA down. Now DNA ligase is going to come through and make all of this one continuous strip. So it would go through and replace my bad taping. Same thing over here. It doesn't really have near as much to do because it only has this one primer over on this end that we did that was different. This whole rest of the thing would all technically be one continuous strand. Over here, though, we have primer, Okazaki fragment, primer, Okazaki fragment, primer, Okazaki fragment. All of these would need to be glued together on this lagging strand. So that's what DNA ligase is going to do, make it all one solid piece. So DNA ligase glues them together. And then we'll go to the back. DNA polymerase 1 would come through. It would make sure that all of these primers that we had laid down were replaced back with DNA. So not only does it have to make sure that the U's are changed back into T's, but it also does have to change basically the whole nucleotide because they have a different sugar. DNA has deoxyribose, RNA has ribose. We did not talk about telomerase. We're not going to worry about that. And then we know that DNA gyrase would come through. We now have two separate DNA strands. They look identical pretty much to the parent strands that we had from the very beginning. Now we just have two of them. DNA gyrase would come through and wind these two strands up into their double helix forms. And then that would be it. On your paper, make sure you label things. Most of the labels are on here. You know the parental strands are on the outside. We always build DNA on the inside parts of the molecules. Leading strand's continuous. Lagging strand is the one that has to keep restarting. Once you label one end a 5', you automatically know all the others. Everything's basically opposite. So 5', anything below it, 3', anything on the very opposite end of it, 3', and the very complete opposite corner of the two sides of the molecule are going to be the same 5'. Make sure you just label what's down here. So we have Okazaki fragments, the chunks of DNA in between the primers, leading strand, lagging strand, parental strands on the outside, and I think that's pretty much it. Let's see. The sheet that I have doesn't have all the papers, so I don't really know. But hopefully that's it. Hope this helps, and see you all tomorrow.